Hi guys, welcome to another video by Pure Knowledge. And this video, we are going to try and do a crash course on diagrams in biology. Some important diagrams in biology that is more than likely to come on the exam. Some more frequent than some, but we're not leaving anything to chance, guys. So just to note, there are other diagrams in your book and your syllabus that you should cover your entire syllabus. You should study your entire syllabus. However, this video covers most of the diagrams that you may need for your exam. So let's go into the crash course. But before we go, just to make note that notes for this video, was taken from the Concise Revision course biology book by Antin Dale. If you don't have your copy already, go and get one, guys. All right. So the most common thing is animal cell and plant cells. So cells, very important, guys. You know your cell, and just to note that I may be going a little bit fast. You can always pause the video, screenshot, take your necessary notes if you don't have the textbook, or if you find this medium easier, go ahead and screenshot, guys. So if we look here, it's very important to know, guys, your diagrams and your functions, right? You must know your functions and your diagram. The functions can help you to answer related questions in the exam. So know your function, know to label your diagram. So common, plant, animal cell, very common, glycogen granules, store food, cell membrane, we know it's a semi-permeable membrane or differential permeable layer composed of protein and lipid. Outside the cell controls what enters and leaves the cell, and it's semi-permeable, so that means not everything can go through. Cytoplasm is where most of the chemical reaction take place. It's a gel-like substance in the cell that's made up of 80% water and 20% mineral substances, right? We know the nucleus has 4 different structures. So the nucleus is like a mini cell. It has a cell membrane that's double. It's double, right? We can see there's a double membrane, right? And we can see where it has a nucleoplasm. We have the nucleoplasm, guys, as I stated before, which basically is like the cytoplasm. So it's same gel-like substances, but the word nuclear means nucleus. Just like the prefix nuclear. Sorry, means nucleus, and prefix cyto means cell, and plasm is the gel-like substances. So nuclear and plasm, that's in the center here. We have the chromatid threads, right? And we have our nucleus, right? So we know these 4 structures, make sure all makes up the nucleus, right? And nucleus is very essential for cell division. Then we have the mitochondrion. It's rod shaped, so make sure that when you're drawing your diagram, it's rod shaped. And this is where respiration occurs. You know, mitochondrion is the powerhouse of the cell. Other vacuoles, which are smaller, right? Because of course, animal cells don't need to store a lot of water. So the vacuoles will be smaller. So that's basically the animal cell, guys. Notice doesn't have a definite shape like the plant cell, which has a definite shape, right? It has a cell wall that's freely permeable, and the purpose of the cell wall is to protect and support the cell, give it its shape. Then we have the cell membrane, which basically is the same as before. We have the chloroplast, where is a disc-like shape. So guys, if you're asked to draw this and you're asked to recognize it, you know that it's disc-like. So you make sure to draw it disc-like. Contains the internal membrane, which is where chlorophyll is formed, where the photosynthetic enzymes are, right, to do photosynthesis, right. Then we have the starch grain. Please note that the starch grain store food in animal cell and not in the starch grains store food in the plant cell, while the glycogen granules store food in the animal cell. Guys, I'm going to fast, so see with me if I miss say some words sometime. All right, then we have the vacuole, which is a large membrane bone compartment containing cell sap. right? A solution of sugar, mineral salt, amino acid, and waste product support the cell when turgid and may store food or cell waste. And you notice how it's big because plants don't really move, so they'll have to store water, right? In case of a drought or anything. So their vacuole will be bigger. Then we have the vacuole membrane, then we have the nucleus, we have the cytoplasm, which is the same as before. So yes, guys, this is a basic structure of the animal cell, plant cell. Make sure you know this down to the T and the functions. We move now to another cell that don't get much attention, bacteria cell, which is considered to be a prokaryotic cell. That means a cell that has a lack of true nucleus. So we look here, instead of a nucleus, it has a nucleoid, which is a single long coil loop of DNA control cell activities. And we also see it as a plasmid, which is also a piece of DNA. Otherwise, from that, we have the capsule, which is a slimy outer covering found in some bacteria which protects the cell. So the bacteria is a prokaryote cell that has 3 layers. That's the capsule, the cell wall, and the cell membrane. Now, the capsule, that's why it's really hard to get rid of bacteria, because, you know, if you look at most detergent, they say 99.9% like Lysol, right? Because bacteria is very hard to get because it sticks on the surfaces. That's why the capsule is a slimy outer covering that allows it to stick, right? Otherwise, the cytoplasm remains the same. The flagellum is another thing that's different. It's a long whip-like projection which aids in movement, right? So the flagellum allows the bacteria to move, bacteria cell to move, guys. So please make note of that, guys. Ensure you can identify the difference between all 3 cells. You can tell what one cell don't have and what cell have. Make sure you have that good, guys. All right, then now we have an amoeba, which is a protactist and fungi, really. So they have a true nucleus, right? But this is the most common one. So we have the cell membrane again, cytoplasm, and this one is easy to draw, easy to it look like a foot, right? It look like a foot with toes, right? So we have the cytoplasm, we have the nuclear membrane, that's clear again, guys, with the 4 structures. We have the nuclear membrane and nucleus, and the nucleus is clear with the 4 structures. Then we have the contractile vacuole, we see here, and just note that amoeba has 2 vacuoles. It has the contractile vacuole that contains water, and it has a food vacuole that contains indigested food organism. So this is where digestion is occurred. And most importantly, guys, this outer region is called the pseudopodium, or the pseudopoda, right? And pseudopoda is false foot, so it's an extension of the membrane and cytoplasm. As you can see, the cytoplasm and the membrane is there, right? And it's used to engulf food for movement. All right, guys, so make sure you know your amoeba, make sure you can identify your amoeba, and let's go. Then now we move on to osmosis, and this diagram is normally important because it's telling you how osmosis works. It moves from a place of high concentration to low concentration across a semi-permeable membrane, a differential permeable membrane. So we can see here where dilute solution is 10% sucrose solution, right? And so because it's 10% sucrose solution here, right, and 50% sucrose solution here, we see here as a more higher concentration. So we see the water molecules diffusing through osmosis. So it's diffused through the permeable membrane to dilute the amount of sugar on this side. So it's water here is higher than water here. So it's moving from a place of higher concentration here to lower concentration here. Again, we'll see the same thing for either cells or just another container. So we see here where the bag contains a 40% sugar. So in the bag has a 40% sugar and outside has 10% sugar. That means inside the bag is more concentrated of sugar. So that means that water is of a higher concentration outside and inside the bag. So the water moves from outside the container to inside the bag. So it's moving from a place of high concentration to low concentration inside the bag in order to nullify the sugar content. Same here, we see the opposite. It is sucrose, 40% outside the bag. That means outside is more concentration of sucrose. That means it has a less concentration of water than inside the bag, right? So water will be moving from inside the bag to outside the solution, therefore it will shrink, right? And in this case, the water will swell here because it's moving to dilute the sugar. Same process for animal cell, you know, if the concentration outside here, if the animal cell is in a dilute solution, it means that water will be moving from outside, from a higher concentration, because water outside will be higher, to a lower concentration inside the cell, right? And then this animal cell would end up burst because it doesn't have a cell wall to protect it. So make sure you can demonstrate osmosis using a cell, guys. Then now we have the same thing for the plant cell. However, we see that the plant cell is placed in distilled water. That means that is low concentration. Remember that the cytoplasm is always 80%, is always 20% sucrose. So if it's dilute solution, it means 100% pure water. That means that now the water will leave from a place of higher concentration outside the cell and move inside the cell, right? In order to nullify the sugar content inside the cell, because the sugar content inside the cell is more than the sugar content outside the cell. And there's more water outside of the cell and inside of the cell. So therefore, it's going to move from a place of high concentration to a place of low concentration. It's going to make the cell swell. The word is turgid, right? And then for the opposite, if there is a higher concentration outside of the plant, so if there is higher sugar content or higher salt content outside of the plant cell, that means it would be, so where there's higher sugar content, there will be less water. So therefore, the water therefore now be of a higher concentration inside the cell, then it will be moving out in order to dilute the water in order to dilute the sugar content or the salt content outside of the cell. So ensure, guys, you know this diagram, you can properly label it, right? So let's move, guys. Let's move. All right. So now we have the leaf, basic structure. Ensure that you know the apex, the lamina, the veins. Pretty simple. The petiole, the midrib is in the middle, and the margin. All right, guys. And then now make sure you know this the leaf from different angle. Right, so this is a transverse section. We can see the midrib, we can see the phloem tissue, the xylem here, and we can see them right throughout, right? But this one is more popular, guys. So we know the waxy cuticle, right, is waterproof layer, which prevents water loss during high transpiration, like when the time hot. Right, we have a chloroplast there that is in the mesophyll layer, right? So we need to know all the layers, the upper epidermis, a single layer, lacking chloroplast. Then we have the palisade mesophyll layer, that's where the chloroplasts are, elongated cells contain many chloroplasts where photosynthesis takes place. So this layer, the palisade mesophyll layer, is where photosynthesis really takes place. Then we have the spongy mesophyll layer, rounded cell with air spaces between them, contain few chloroplasts than the palisade layer. So ensure we know all of this, guys. Then we have the lower epidermis, right, where the guard cell is, and the guard cell is right here. That the guard cell is bean shaped, right? The inner wall thickened with the cellulose, and chloroplasts are present in the guard cell. And the space between the guard cells is called the stomatal pore, right? And together they are the stomata, or the stoma. Stoma, singular, stomata, plural, right? So the stomata is guard cell and stomatal pore, and together collectively, it is called the stomata. So the guard cell basically control the opening and closing of the stomata. Then we have the ear space, where air are stored in the cell, in the leaf, sorry. And then now we have the vein with the xylem vessel, the phloem seed tube, and the companion cell here. and the waxy cuticle underneath, guys. So make sure we can know the structure, we know the function of each of these layers. So let's move. Then now we have the teeth, guys. Ensure we can label the teeth properly. Ensure we know that the enamel is the hardest substance in the body, right? Resistive, it's really resistant to decay. So, however, it can be dissolved by acid produced by bacteria in the mouth, and basically the enamel protect the tooth. That's why we have to brush our teeth, guys. Ensure that we are good. Then now that's the first structure and the outer structure. Now the middle structure is the dentine, which is bone-like. So remember I said enamel is the hardest substance. I mean it's harder than bone. So the dentine is actually bone-like, containing channels of living cytoplasm from cells in the pulp cavity from a bulk of the tooth. And then we have the most sensitive layer now, which is the pulp cavity composed of living cells. So basically, when our teeth rot, guys, it has to bore through the enamel, it has to bore through the dentine, and then the pulp cavity. Where we start to feel little pain, it's actually the dentine. When we start to feel the immense pain, it reaches the pulp cavity, guys. Other things we need to know, the blood vessel supplying the teeth with food and oxygen, and remove carbon dioxide. We have the gum, right? We have the jaw, the nerves here in the middle here, the cement, right here, the fiber, right here, and the fiber allows for a shock it's basically a shock absorbent, right? So any little bounce your teeth might get, it can withstand it, right? So yes, guys, these are the basic structure. Make sure you know the overall thing. So the top above the gum is called the crown, right? The part that you can see is the crown, and below where you can see is the root. So make sure you know all the structures, guys. Then now we have the digestive system, right? So we know we get like a rundown. So you know you eat food, food goes in the mouth, and it's breakdown by enzyme, right? Enzymes. So it mix with the food basically, binds at the active site, right, guys? Then you swallow it through a wave-like movement called peristalsis. You know, chewing. Another name for chewing is mastication. So you say you masticate your food. The process of taking in the food is called digestion. And when we start chew it, is actually, or masticate it, is when digestion actually starts. So we start break it down so that the enzymes can bind. But apart from that, guys, we need to know all the structures here, the pharynx, the throat, the epiglottis, cause the trachea. Closes the trachea during swallowing. So when you're swallowing your food, the epiglottis prevents it from going down your windpipe. So sometimes food do escape and you cough. So that is the epiglottis really, especially if you're eating in a hurry. Then we have the esophagus, transport food to the stomach. The stomach produces gastric juice, like the hydrochloric acid and the pepsin, right? Digestion of protein begins, and we know that pepsin is what digests protein. Then we have the pyloric sphincter muscle, which relaxes periodically to release the food from the stomach. Pancreas produces pancreatic juice, and those include the pancreatic enzymes such as amylase, pancreatic trypsin, pancreatic lipase, all right, just to name a few. So ensure you know your enzyme, guys, and the proper pH in the body. Then now we have the part of the small intestine, which is the duodenum and the ileum, right? And the small intestine is where absorption of the food and assimilation of food occur. Absorption, and it goes into your cells, and assimilation when it is used by the cells to produce energy through respiration. So let's know bile, liver produce bile. We know the diaphragm separate the respirator from the digestive system. Bile duct, pancreatic duct, cecum, appendix, no function, anal sphincter. So guys, rectum, colon. So guys, like, ensure you know this diagram, you can label it. You know the different enzyme in the specific parts of the body, and we move. So now we talk about the villi, and the villi basically help the villi is found in the small intestine, and the villi basically help to increase the surface area so digestion and assimilation can take place properly. So it's really increasing the surface area so assimilation can take place, or we call it absorption can take place, and then assimilation take place. So basically, the villi helps with the absorption of food, right? And that is from the small intestine. So we need to know to label the villi, or the villus in this case, because it's one. It's one, so singular villus. So you have the epithelium, wall of the villus, one cell thick and one cell thick, right? So that it allows for assimilation. It has to be thin so that assimilation or absorption can take place. I keep on saying assimilation, but it's really absorption takes place, because assimilation is when the body cells actually start to use the nutrients, right? So you have the blood capillaries, ensure you can label that well, guys. Absorb water, water soluble substances. Make sure you can label this. Make sure you know what the lacteal is. Lacteal absorb fatty substances. Make sure you know that this is that. We know the venule is a hepatic portal vein that takes water soluble substances to the liver, right? And hepatic portal vein takes food from the liver to the stomach, right? Lymphatic vessel takes fatty substances to the blood before entering the heart. So guys, make sure you know all of these. Make sure you know the microvilli, the goblet cells secrete mucus, epithelial cells. So this basically is a little piece of epithelial cell, and we are labeling that. So guys, let's sure we know these structures well. Then now we move on to the respiratory system. Ensure you know the larynx, the voice box, you know, the trachea, you know, a hollow tube with rings of cartilage in its wall, which keeps it open. Let's know the left lungs, left bronchus, you know, tube with rings of cartilage, right? The heart here, so the ribs, which protect the lungs, diaphragm, we separate the respiratory system from the digestive system, right? So we look at, this is the alveoli, right? We look at the pleural membrane, which surrounds the lungs, right? The outside of the lungs are pleural cavity, which is filled with pleural fluid, right? Airtight and attaches the lungs to the rib cage and the diaphragm. So let's show we can label all of these. The bronchioles, right? Hollow tube branching throughout the lungs, lined with moisture, internal intercostal muscle, external intercostal muscle, guys. So ensure we can label these diagrams well. All right. Then we have the villi, right? So the villi now is, this is basically showing, right, how blood is oxygenated using the alveoli. So what is the alveoli? The alveoli basically increases the surface area in the lungs so that respiration, so that exchange of gases can properly take place. So oxygen and carbon dioxide can exchange properly, right? So we can see the capillary from the pulmonary artery, right, carries deoxygenated blood, and pulmonary artery is the only artery that carries deoxygenated blood, right? The alveoli is thin wall, provide a large surface for gaseous exchange, right? Has to be thin, just like how the villus is thin. Anything that is facilitating exchange, right, it has to be thin, right? So we see the villus, which is singular to the alveoli, all right? So same different, guys. We have the network of capillaries surrounding the alveoli, bronchioles, and then we have the capillary to the pulmonary vein, which carries oxygenated blood. So deoxygenated blood is coming in the alveoli and is getting oxygenated. And when it gets oxygenated, it goes out, and the pulmonary vein now carries it to the rest of the organs or the rest of the body. And then they are distributed. So when we're going to circulatory diagrams, we'll see better. But make sure you can label this diagram, guys, and know what it's about. Then, however, the same diagram basically, but different structure. Where we see deoxygenated blood. I remember, guys, deoxygenated blood is always on your left, and your right is oxygenated. If you didn't, I want some tips on that. So it's the same diagram, basically, guys. Explain the same thing. Only thing it's explaining is that how the exchange of oxygen differs. So we see more clearly now that here would be carbon dioxide, then the carbon dioxide diffuses out, and we see where the oxygen, so when it's exhaled, we see how the carbon dioxide moves out, When he inhale here, we see oxygen move in, and we see how it now makes the blood oxygenated as it goes to goes through the pulmonary vein to the rest of the body. Make sure you can label this. Is a layer. This is a layer of moisture. So moisture basically help the oxygen to be dissolved, so it can be better diffused into the body where necessary. We have the red blood cell and the wall of capillary. One cell thick, one cell thick. See, one cell thick means that it's thin enough to facilitate diffusion, guys. So yes, guys, let's look at this video. Let's look at the image and let's get this done. So get your images done so that you can get your label, your diagrams labeled properly for exam. All right, then we have the fish, which most people ignore. So let's look at the fish, guys. Let's bring it up a little bit. Let's look at the lamellae, right? The gill lamellae, right? So let's can label it, right? So this provides a large surface area for gaseous exchange. So the lamellae would be like the alveoli in human. The blood vessel will be here. The gill racers filter blood from the water passing over the gills, right? And the gill bar is an arch of bone, guys. So let's ensure that we can label this well. This is the fish, which is overall diagram. We're not taking any chances, guys, to look at the gill raker inside, right? We look at the operculum, which is the little dot lines here that goes around, right? Then we have the 4 gills at each side of the head, and we have the gill bar. So we're not taking any chances, guys. So let's look at these diagrams and get that one. Let's go, let's go. Then now we have possible changes based on diseases, right, that will affect the lungs. So we look at when you smoke, especially marijuana, you have excess mucus in the bronchi, and then we see how the mucus now will stop. We see how a healthy mucus is free. We see how the mucus will clog the airway, right? So therefore it will allow us not to breathe very well. Look, emphysema, right, affects the alveoli. So you look how it look when it's healthy, how it look when it breaks down, guys. So let's go. Now we move on to the red blood cell, guys. We must know to differentiate between our red blood cell and our white blood cell, guys. Red blood cells is biconcave, right? It has no nucleus. So therefore it lives for around 120 days, 3 to 4 months. It basically don't have any nucleus, so it have a large surface area to transport oxygen, right? So make sure we can label this, guys. All right, it has in hemoglobin, which makes the blood pigment red, right? And hemoglobin is a type of protein. Cell membrane as usual. Then we move on to the white blood cell, which are 2 types of white blood cell, lymphocyte and phagocyte. Lymphocyte basically has a large nucleus because lymphocyte need a nucleus because it now produces antibodies to fight diseases, right? So it will need a nucleus, a large one. So we can see cell membrane, a large round nucleus, and non-granular cytoplasm. Now for the phagocytes, and these are phagocytes, so your lymphocytes and phagocytes make white blood cell. They are lobed nucleus. They have a smaller nucleus, which is lobed. Make sure you have that, guys. Make sure it's lobed, but if you see it, make sure you can identify. Once it's lobed, it is a phagocyte. Lobed because the phagocyte main job is to engulf bacteria, as it shows you how it's engulfing bacteria here in this diagram, where it forms a circle around this bacteria, and then it engulfs it here and it destroys it here. So basically, because it engulfs bacteria, it needs more space to do that. That's why it has a smaller lobe nucleus, guys. So make sure you can identify and draw if necessary, because these are simple diagrams. All right, then we have the heart, guys. Ensure you know our hearts well. To ensure we can label the anterior vena cava is coming from the head, so it carries deoxygenated blood from the head, right? And then we have the posterior vena cava, which carries deoxygenated blood from the body. So we see where deoxygenated blood is coming through 2 sides here, right, guys? So we see that deoxygenated blood is coming from here and it's coming from here, right? Then we have the artery that collects those deoxygenated blood. That is the right artery. Remember, guys, on the right, on the left is on the right, right? So deoxygenated blood is on the left, and the set of organs in the heart that deals with deoxygenated blood is the right. So you can see here the right atrium, right, the right ventricle. So the right ventricle, the right atrium deals with deoxygenated blood, guys. That's a trick, right? So deoxygenated blood is to your left, and the organs that deal with it is to your right. So right, and it's on your left. So that's a whole trick, guys. So then the right atrium collects deoxygenated blood, right, from the head, and we see it there. And then the tricuspid valve prevents blood flowing back to the atrium when the ventricle contracts. So the valve prevents any backflow. So the tricuspid valve is on the left-hand side, guys. Then we have the right ventricle, pumps oxygenated blood to the lungs. And we can see here, it pumps the blood back up to the lungs, right? And the pulmonary artery will carry the deoxygenated blood to the lungs. Now, the pulmonary artery, guys, is key to note, is the only artery that carries deoxygenated blood in the body, right? Other artery carries oxygenated blood, right? So then now we have the aorta, which is the main artery. That I call the vena cava is the main vein. Aorta is the main artery. So it carries oxygenated blood to the body. So let's look at the process. So it's the same one here, same process here. On the right, where we see the left atrium collects the blood from the lungs, right? And then the pulmonary vein will carry the blood from the lungs. So what happened now, guys, is that after the right ventricle pump the blood to the pulmonary artery, right? The pulmonary artery carries this deoxygenated blood to the lungs, right? And then this deoxygenated blood, right, that is in the lungs, now it gives the blood to the pulmonary vein. So the pulmonary artery gives the blood to the pulmonary vein. Then the pulmonary vein carries that oxygenated blood from the lungs, because when the pulmonary artery carries deoxygenated blood to the lungs, it will get oxygenated, as we see with the alveoli, right? So remember the alveoli, we see where it's getting oxygenated, where the carbon dioxide is moving out and the oxygen is moving in. Correct, that is how. So after it done that, it gives it back to the pulmonary vein. The pulmonary vein now goes some boops and push it down here. And then the semilunar valves prevent the backflow into the ventricles when they relax. So the semilunar valve, very important, guys. And the bicuspid valve does the same function as the tricuspid valve. So it prevents it from going into the atrium in the left ventricle side, right? So the bicuspid valve, just to be clear, the bicuspid valve prevents backflow into the atrium when this left ventricle contracts, right? The tendons prevent the valve from turning inside out. Yeah, that's some basic common knowledge there. Then now we can see where the left ventricle is thicker than the right ventricle because the oxygenated blood has a longer distance to go in the body. So the right ventricle is just pumping it to the lungs, one job. But the left ventricle will be pumping it to the entire body. So it's pumping it to the aorta. And then the aorta will then carry it around the body. All right, so remember, guys, pulmonary. So the pulmonary vein collects it from the pulmonary artery and then push it down. The valves ensure it stays on the right path. And then the left ventricle, which is thicker because it has a bigger job to do, transporting it to the entire body, pushes it right up. And then the aorta carries it around the body. So I hope that makes sense, guys. Make sure you know your diagram. You can label your septum, your cardiac muscle, and you can properly state where the blood is going. All right? Same thing here, guys. Right, so same difference. If you understand the first diagram, we see the pulmonary artery carrying it to the lungs, right? We see the right atrium, it's collecting it, but right here, we see the right atrium is collecting it here, and then we see it's collecting it from the body, the vena cava. So the only thing different is that this diagram don't have a anterior vena cava, right? So it's not taking it from the head. It's only the posterior vena cava showing in this diagram because it's taken from the body and it pushes up to the aorta, to the atrium, sorry, and the atrium collects it here and then pushes it to the pulmonary artery, and the pulmonary artery takes to the lungs, and then the pulmonary vein takes it to the left atrium, right? And then the left atrium collects it, and then it pushes it to the left ventricle, and then the left ventricle pushes it to the pulmonary, sorry, pushes it to the aorta, and aorta carries it around the body. So we can see, guys, the same thing, same context. So make sure we can label that any way it comes, guys. All right, this is basically transection, transverse section through a root of a dicotyledon showing arrangement of xylem and phloem. So make sure you can label this pretty simple root here, extension of a single epidermal cell cortex in the middle here, packing tissue composed of spherical and unspecialized cell, endodermis, single layer of cell, vascular cambium constantly dividing cells between xylem and phloem, forming xylem inside and phloem on the outside. And we have the xylem and the phloem here, guys. So make sure you can label that if it comes. This is very important, guys. The guard cell, they're showing how the guard cell works. So the guard cell taking water by osmosis and become turgid, therefore it opens, right? We see the thicker inner wall doesn't stretch. So that's important, guys. You can read that, you can screenshot and read, right? But important to know, it becomes turgid and it opens. When it closes, it becomes flaccid. So when the guard cell closes to prevent water loss in the plant, it actually becomes flaccid to not allow the water content to release from the plant. Then now we're moving on to our urinary system, our renal vein, our left kidney, our artery, you can see the posterior vena cava and aorta urethra, which carries urine to the bladder. So we see it's connected to the kidney and the bladder, right? And then the bladder stores the urine and the urethra carries the urine out of the bladder through the penis. You have the sphincter muscle here. So make sure you can label all of this, guys, if it comes. Then now we have a longitudinal section through a kidney showing the position of a nephron. So we see the nephron here. Guys, make sure you can label that if it comes. Collecting duct here. To the renal artery, renal vein, pelvis, cortex, urethra, pyramid, inner part of the medulla, medulla inner region. So make sure you read that, guys. And the glomerulus, right? Never can pronounce that word, but the glomerulus, right? Sure, get that right. It's a cluster of capillaries at the end of the arteriole. So make sure you can spell glomerulus if you can't pronounce it like me. And then now we have the Bowman's capsule. That's easy, guys. It's always a cup-shaped structure around the glomerulus. So we can see. We see a better diagram here, but we can see the cup shape. All right. So make sure you know this, guys. Then make sure you know the nephron itself, right? And it's very important to know our functions of the nephron. So the nephron is basically about reabsorption, guys. It's about reabsorption and filtration, right? So basically the blood comes in here, guys, and then it goes to the glomerulus, and the glomerulus basically, in the glomerulus, the blood is forced out into the Bowman's capsule, guys. And there now from Bowman's capsule, we have ultrafiltration. So this is where the blood is filtered, and then they normally filtrates with the glucose, amino acid, hormones, vitamins, water, salt, and urea. All right. And then it goes down, right, here, around the medulla, cortex medulla, around here, right? At B, we see that there is selective reabsorption. So here now, guys, if the body is short on water or short on mineral substances, the kidney would reabsorb back the things that it filtered, right? So the things that it considered waste, it will reabsorb it back if the body needs it, guys. That's very important. All right. So there is selective reabsorption. Selective meaning based on what the body wants. So it selects what it needs. So all of those things that it filtered, it can reabsorb it again, guys. Then now water is absorbed through osmosis, you know all of that, and then the substance will be active transport, which is energy, across a concentration gradient. Then now all of this will be down to B, right, guys? And then label it nice for your B, 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 B. And then now at C, we can see where filtrate is filtered and then it can be reabsorbed as well in C. So some reabsorption do take place in C as necessary, but most of it would have been getting rid of the substances. So make sure we can identify it. So we know that the loop of Henle is here where selective reabsorption takes place. We know that the first convoluted tubule, right, is where most of the ultrafiltration would have taken place. The second convoluted tubule is where selective reabsorption would have taken place. So make sure we can label all of these things, guys. Very, very important. Guys, pay attention to this so we never might know what pops up. Then now we have the skeletal system. Pretty straightforward, guys. You can just take a screenshot of that and show you know all the bones in your body, where they are located, guys. And make sure you know the joints in each section of the body. All right. Then now we have the nerve system. So we have three types of nerve system, basically. We have the sensory nervous system, where this is the receptor that the skin that receives. So yes, and the place is warm or the place is hot, right? Not hot enough for a reflex action, but it's picking up something at the time it's cold, right? Then it sends it along the dendrite. Right, so you have a receptor dendrite, then we have the myelin sheath, is a fatty insulated insulation that protects the nerve fiber here and speeds up transmission of impulses. So we know that all of these things are the myelin sheath. The little grooves that are in between here are called the nodes of Ranvier. So make sure we know that. And then when it reaches the cell body, it is the axon, because the axon now is from the cell body, right, to the synaptic knobs. And the direction flowing this way. Then now we have the motor nerve, right? We see the dendrite here, which are finger-like projections of the nerve, where transmission takes place, where this is where cells communicate and talk to each other, right? So the communication will take place between the dendrites and synaptic knobs. Note that they do not touch, they never touch. What the message does is jump across, right? Jump across, then we have the nodes of Ranvier, same, we have the myelin sheath, same, we have the axon. All of this would have been the axon because the axon is from the cell body, right? And this is a cell body, right? Before the cell body is a dendrite. You can see the dendrite here, but here the dendrite should have been on the cell body for the motor neuron, right? And then it goes to the effector. Effector is a muscular gland. Then now we show you how it works. Overall, sensory neuron, PNS, right? That's the peripheral nervous system, goes this way, receptor, and then the synapse is the space between the dendrite and the synaptic knobs, where this is the space where the information will jump across. Then we have the relay neuron, does not have a myelin sheath. Make sure you can indicate that if needed, and it jump across here, and then go here to the effector, guys. So very important to note all of these three, guys. Make sure you understand it, you can draw them and you can identify them if needed. Then now we have the brain, pretty much straightforward. Right, guys, we can just pause this and read these things. Very straightforward. Cerebrum, movement and control balance. Medulla, controls automatic activities. Cerebrum divided into regions, different functions. So all of this is the cerebrum. Frontal, decision making, intelligence, guys. So make sure you're aware of every little thing. Then we have the eye, guys. Just pause the video and read all of that, guys. Very, very important, guys. You know all of this, what it does. Very, very important. The eye is critical. The eye came a few years ago, right? Not very far. Eye is very critical, guys. So make sure we know our eye well. It's a great diagram of the eye, by the way. Ensure we know what images are formed, just in case, right? We know the cornea refracts light rays. You can see it here. You know, the lens refracts the light rays, focusing them on the retina, right? And then we see an image of the object is formed on the retina, stimulates the light sensitive cell. The image is inverted, reversed, and smaller than the object. And the optic nerve carries the impulse to the brain. So guys, make sure we can draw this label, understand it where needed. All right, make sure we know accommodation process, guys. Accommodation, very important. Accommodation speaks to when we're looking at something far away and then we change it and look to something near. So the eye has to accommodate, right? And make sure we know the processes. This is a process of focusing distant object. The cerebral muscle relaxes, right? Suspensory ligaments are pulled tight and the lens is pulled thin by the suspensory ligaments. And then we can draw it if we need to or identify what is going on. It's very, very important, guys. This is important in drawing and in explaining a regular question. Same for B, opposite, cerebral muscle contracts, circumference decreases, suspensory ligaments flatten, the lens allow the spring to bulge into shape, guys. So we can see it bulge into shape right here. So guys, pay attention to this diagram. Very, very, very important. It's a common eye question. So let's be ready for it. Then now we have controlling the amount of eye and light entering the eye, which they're pretty basic. They'll give you these two and then tell you what's happening here. You know, it's dim light, here it's bright light. You know, so very, very important, guys. Then we have the skin. Very important that you pay attention to all of the layers here, right? And you can, you know, just pause it, read it, see what's up. It's very, very straightforward, guys. Make sure we know the processes of cooling in high temperature, what the skin does in high temperature in response to a stimuli versus when it's cold, the ear stand up, when the ear lie down, and what the blood vessels do, right? Let's know that, guys, all right? That the sweat gland produce sweat, right? The blood vessel come near to the skin, warm it up, all of those things there. Make sure we know what's popping there. Then no germination, guys, very important. They can ask you to label, right? So this is the heliomeres right here. The micropyle allows water into the seeds for germinate, and the water would activate the enzymes inside the seed for growth, right? And the testa is a tough protective coat of the seed. We see the plumule here, the radicle here, micropyle, which would have been here, the testa same way, and two cotyledons, right? That stores food, right? So make sure we can also explain when it's germination process. Normally, them give you 5 of these things and you have to explain. Make sure you can label it properly, where it emerges, and make sure you know that when the seed is in the soil, no photosynthesis takes place. Photosynthesis only can take place when the leaves are out, right? So while it's in the soil, it really and truly relies upon the food stored in the two cotyledons. Yes, guys, make sure we know our reproductive systems, right? Very straightforward, guys. Make sure we know fallopian tube or oviduct or functions here, vagina, the cervix, the uterus. Make sure we know that. And if case they ask you to show how fertilization occurs, they ask that one here, you know, the sperm will travel here and goes to fertilize the egg through the fallopian tube there, guys. So pretty straightforward, guys. So you just revise right there, here and now. Then we have the male. Ensure you can know the vast difference at the sperm duct that carries sperm to the urethra. It's been a while since I saw the male, so pay attention to it. The epididymis, which stores sperm. Then the penis, which is very evident, has connective tissue with many blood spaces around the urethra to becomes erect during intercourse, deposit sperm into the vagina. The urethra, we know what that is already, guys. Carry semen outside of the body, because we wouldn't say urine in this case, because we're talking about the reproductive system. So urethra, it carries both, but it's a reproductive system we're talking about. So we use context, guys. The testes, we know the testes produce and secrete sperm while the epididymis stores it. The sac, the foreskin. The seminal vesicle, and the prostate gland. The gland that secretes fluid which mixes with the sperm to form semen. The fluid stimulates the sperm to swim. So urethra from the bladder. So guys, just get familiar, all right? Get familiar that I want to get that one. All right, then now we have the menstruation cycle. We know that first, this is menstruation occurring here. And when menstruation occurs, this is the uterus lining tearing down. Then now the body produces estrogen, and estrogen basically help to thicken the uterus lining back, right? And that normally occurs up to around 14 days. Then progesterone takes over, right? And progesterone causes the lining to remain thick and get thicker, so that if the egg is deposited into the uterus, it can attaches itself and the environment is right. Because blood is needed for oxygen and food for the baby to survive. So that's why these hormones make the cell wall, make the uterus lining thick. Then now, if there's no implantation, then menstruation will occur. So there's a decrease in the progesterone causing the uterus lining to become breakdown. All right, and this is just showing the hormone level, guys. Same thing as I explained. So this is showing the hormone level. So estrogen would rise, causing the wall to get thick after menstruation. And then progesterone would rise, and the estrogen would decline because the progesterone is making it getting thicker, right? Estrogen is secreted by the Graafian follicle. It's like a round thing here, which we'll see here. Normally it's produced here. And the progesterone is secreted by corpus luteum, which is normally here. All right, guys, and then this now showing the egg maturing inside the Graafian follicle. And then after 14 days, it would burst out. All right? So if there's no fertilization there, then nothing will happen. All right, so the corpus luteum develops, and then it degenerates and the cycle occurs all over again, guys. So that's menstruation in terms of diagram in a nutshell. Make sure we are familiar, guys. Then now we have the developing of human embryo or fetus. Again, guys, pretty straightforward. You know, the placenta provides a lot of services for exchange of substances between the mother and the child. We have the thickening uterus with rich blood supply, capillaries, guys, which you can see that here. And we know food and oxygen from the mother's blood. In the uterus lining, it goes to the child. Carbon dioxide is diffused from the embryo into the mother's blood so the mother can get rid of the waste for the baby. Villi increases the contact between the placenta and the uterus lining. Make sure you know what the amnion is. Protect the baby, the amniotic fluid that's inside the amnion, right? The plug of mucus that is here, the vagina, which is here, the umbilical cord, which carries dissolved carbon dioxide and other waste away from the embryo, umbilical vein, which carries dissolved food and oxygen to the embryo. Very important, guys. Embryo fetus, umbilical cord, muscular wall. So ensure we have all that lot, guys. Then now we have the flower. We have seen the male, the flower asked a few times. I think this year we can pay very much attention to it. But for general purposes, the flower is very, very important. All right. So we have the stamen, which is the male part, and the stamen consists of the anther and the filament. Right. Then we have the carpal, which is the female part, which consists of the style, the stigma, the ovary, the ovule. So let's read that, guys. It's pretty straightforward. Right, so make sure you're really, really, really familiar with this. All right, guys, if you don't have a book, you can always pause the video, as I said, and get that done. All right, then we have, wind pollination. Suppose them ask you, look at this diagram, identify what type of pollination it is. It's wind because of how the structures are stay right here. The structure them stay right here. Yes, guys, this is just a quick example. Then now we look at fertilization in flowering plants. We look at the pollen grain go on the stigma, right? And we see where the pollen grain lands on the stigma. And the pollen grain with two nuclei and a tube nucleus on its tip, which would have been right here, right? Goes down through the style by secreting digestive juice, because digestive juice will have to break, so it have to break down, get the things out of the way. So it can go down. Then now we have the stigma, right? And we can see the stigma here at the top. So then the pollen grain would just go right down, and then it goes down to the ovary wall through the micropyle. Remember, the micropyle is a small little hole right there for the ovule and burst. So it goes to the micropyle of the ovule and burst. So it goes through the micropyle and burst it. I can see a little rupture here. and a tube nucleus degenerates, and one male nucleus fuse with the female gamete to form a zygote. The other male nucleus and two polar nuclei fuse to form endosperm. So guys, we can pay attention to this. I haven't seen this in a long while, but just to take precaution, guys, we can always look at it. All right. Then now we have the life cycle of a mosquito. So we know it's an egg, then it turns a larva, right? Then it turns a pupa, then it turns an adult fly, and these are the sections of it, guys. So we can look at the larval stage, mosquito, just to be, and this works of fly as well. And that's it, guys. So hope this video was helpful, guys. Hope this crash course was very helpful. It's a crash course of diagrams, so I may have moved fast a bit, so you can always pause the video where you like and get it done. So all the best for your exam, guys, and I will see you on the other side of our next video. So later, guys, all the best.